Good morning. Please answer your attendance. $50. $67. $78. $82. $86. $91. $98 absent. $46. Okay. We got to finish two more pathways, citric acid cycle and HMP shunt pathway. Unlikely to finish it today, I guess. But I'll try and briefly finish with what is possible. We were dealing in the last class with the significance and what that Rappaport Liebering cycle is all about. It's a shunt pathway. Anybody would be able to tell the significance of a Rappaport Liebering shunt pathway? Any? Any significance you know of? Which is that product formed? 2,3-BPG. The moment you talk of Rappaport Liebering cycle, you need to talk of 2,3-BPG. 2,3-BPG in itself had come as a question where its significance needs to be written. But now it's not necessary that it comes only in Rappaport Liebering cycle, though that is where that is formed. But the chapter on hemoglobin also talks in detail about 2,3-BPG and how the affinity of hemoglobin to oxygen is reduced, and thereby it's able to release that oxygen even when the partial pressure of oxygen is low, and that is a significant feature when not on normal conditions, but when conditions of decreased oxygen is present. And then a few factors associated there. So we were dealing with actually the breakdown of glucose. We have broken it down to first to pyruvate, and then the conversion to by the dehydrogenase complex to acetyl-CoA, which can be considered the stage 1 of the entire process. Stage 2 is largely to do with TCA cycle and how this acetyl-CoA by condensation and then an 8 or 9 step process, more called a cycle, because where we begin with, we end with that same product. So oxaloacetate being that starting product along with acetyl-CoA, and we eventually get oxaloacetate back in the cycle. So therefore, more than a pathway, this is called a cycle. So stage 2 of it is the oxidation of acetyl-CoA, where a lot of NADH and FADH is produced, not exactly ATP, but when it enters into the electron transport chain, they translate into the formation of this important product called ATP. Stage 3, largely, we are dealing with that formation of that electron transport chain where in oxidative phosphorylation, there is the most important product being formed, and that of ATP. So what we had until now was largely taking place in the cytosol. When we reach the TCA cycle, and from there on, even the production of ATPs are all taking place in the mitochondria. So site is where it is important. So we already spoke of that conversion of pyruvate to acetyl-CoA, pyruvate dehydrogenase complex. Though it is not a single step, it is a combination of quite a few steps. We call that a dehydrogenase complex enzyme because there is multiple steps that are taking place, but eventually what we need is acetyl-CoA, and that's where we begin our TCA cycle. Now, in terms of examination point of view, we generally don't get a question where there is a complete oxidation. So it is either a glycolysis or a TCA cycle. So in that context, we use those pathways or cycles in specific manners, but understand it in a context of stage 1, 2, 3, or maybe a complete oxidation process. So this cycle, we would be having about 8 or 9 enzymes that are important, all of them very, very critical. We have a list of steps which are producing NADH or FADH, which eventually gives our the product, or the most important one, the energy. There is one step which produces a substrate level phosphorylation, but it is not in the form of a adenosine triphosphate, but a guanidone triphosphate, which again translates to ATP itself. So which are those 8 or 9 steps that we need to know? So we are beginning with acetyl-CoA, where the first step is a condensation reaction where oxaloacetate combines or condenses with acetyl-CoA in the presence of a citrate synthetase to give you citrate. Citrate synthetase is the enzyme which is giving you the first product, citrate. This citrate undergoes a 2-step dehydration and then a hydration, largely done by one single enzyme, which is aconitase. 2-step again, but you can use it as a 2-step or a single step. It really will not matter much, but if you are interested, you can say that it is once dehydration and then a hydration back, both done by that aconitase enzyme, where citrate is first converted to cis-aconitase, again another aconitase enzyme by the process of hydration, will convert it to isocitrate. Even if you have written citrate to isocitrate, biaconitase enzyme will not matter. Though we put it as a 2A or a 2B step, it doesn't matter. You can still convert it into directly a isocitrate. Now, isocitrate on step 3, you don't need to know this oxidative decarboxylation, hydration, dehydration. Not necessary that you write it in the examination, but if you want to know in the structure wise and understand it, it becomes much easier if you learn it that way. So we are talking of step 3, where this isocitrate by oxidative decarboxylation, simply losing out a carbon dioxide, isocitrate dehydrogenase. Moment the enzyme has got a dehydrogenase, you can translate that into some amount of ATP being produced. We had that even in the glycolytic cycle, where dehydrogenase, the moment you say isocitrate dehydrogenase is the enzyme which is converting isocitrate to alpha ketoglutarate, there is formation of NADH. And for the formation of NADH, obviously we need NAD. NAD is converted to NADH, where isocitrate dehydrogenase is the enzyme. It's a decarboxylation reaction basically, where isocitrate is converted to alpha ketoglutarate. Back to back, you have 2 dehydrogenase enzyme, and this is largely a complex again, which is a 2-step reaction. Again, you don't need to know the details, but simply understand that alpha ketoglutarate, with the help of an alpha ketoglutarate dehydrogenase complex enzyme, again gets converted to succinyl-CoA, but since there is a dehydrogenase complex enzyme, it again obviously produces another NADH. So 2 steps back to back produces NADH in this reaction. So alpha ketoglutarate, the enzyme is alpha ketoglutarate dehydrogenase complex. This is an oxidative decarboxylation, a 4th step in the citric acid cycle, which is giving you succinyl-CoA. The next step, where succinyl-CoA is converted to succinate, is by the enzyme succinyl-CoA synthetase or succinyl-CoA thiokinase. Now this enzyme name is different in different books. Now it is up to you, you can use any of it. Thiokinase can sometimes be helpful if you use, because the kinase again comes into play. And whenever you saw it in glycolysis as well, the moment the kinase came, you had a production of ATP. Here in this case, succinyl-CoA synthetase enzyme or thiokinase enzyme produces a GTP. Again, a substrate level phosphorylation, the only step in citric acid cycle which produces a substrate level phosphorylation. Rest of them are all oxidative phosphorylation in the form of NADH or FADH. So succinyl-CoA, with the presence of the enzyme succinyl-CoA synthetase or thiokinase, produces succinate. Succinate dehydrogenase, again another enzyme, succinate, will be converted to fumarate. Dehydrogenase enzyme obviously either produces a NADH. In this case, the 3rd step, which is producing oxidative phosphorylation, produces a FADH formed from an FAD. So FADH, succinate dehydrogenase, is the enzyme which is giving you a fumarate. Now this fumarate, in the presence of enzyme fumarase, will convert it to a malate. It is a hydration step where a molecule of water is added, where fumarate is converted to malate, and the enzyme is fumarase. Malate, in the presence of another dehydrogenase, the last in that reaction, in step 8, malate with malate dehydrogenase produces oxaloacetate, where we all began from. So malate, in the presence of that last dehydrogenase, which will again produce an NADH, gives oxaloacetate. This oxaloacetate is the one which did the condensation of acetyl-CoA to produce citrate in the first step. So we began with oxaloacetate. we are ending with oxaloacetate. That's why this is more of a cycle than pathway. Now, 3 of the steps had dehydrogenase enzyme, isocitrate dehydrogenase, alpha keto glutarate dehydrogenase complex, malate dehydrogenase, 3 producing NADH. One of the other dehydrogenase, succinate dehydrogenase, produces an FADH, and thiokinase or succinyl-CoA synthetase is the enzyme which is producing a substrate level GTP in the step. Largely a 8-step reaction which gives you the TCA cycle. So a few additional things that you probably may need, but if you have written the cycle, not necessary with that structures, don't bother about the structures, just know the names of the substrate and the product with the name of the enzyme, more than sufficient when you're needing to explain a TCA cycle. How do you remember it? I think it is up to you how you want to remember it, but the 8-step reactions can simply be first remembered in the name of a form, in the format of the substrate and product: citrate, cis-aconitate, isocitrate, alpha-ketoglutarate, succinate, fumarate, malate, oxaloacetate. If you know this part, you know the skeleton. Now you just need to fill in the names of the enzymes. Now they are very, very simple, because you know 3 of them produces an NADH, 1 produces an FADH, and 1 produces a GTP. It's up to you how you want to remember it. I leave it entirely to you, but this is probably a skeleton way of trying to remember what the substrate and products are, and then fill in the names of the enzymes. It will become very simple. A couple of enzymes have got other names, like I told you, succinyl-CoA synthetase, coenzymase synthetase. Some of authors prefer succinyl thiokinase, and that sometimes is easier to remember because it's that GTP concept which is coming up there in that step. So 3 NADH plus an FAD plus GDP, phosphide and acetyl CoA, will eventually give you 3 NADH, 1 FADH, 1 GTP, CoA, and carbon dioxide. So this is what eventually happens in a TCA cycle. That's low part of it is not visible though, but it is producing 3 NADH, 1 FADH, 1 GTP, CoA, and carbon dioxide. So if you remember, the first 3 is more than sufficient. How many NADH, how many FAD, and GTP would be summarizing this entire 8-step reaction in TCA cycle. I'll skip this part entirely. This is just explanation of those 8 reactions and what happens in each of those reactions. I don't see a necessity that you should know the real chemistry behind it, how it combines, what this coenzyme A does to the, and how it modifies the structure and gives you the product. If you know the basic names of the substrate and product, more than sufficient with the name of the enzyme and where it produced the our oxidative or substrate level phosphorilizations. Each of those reactions can be explained. Now, we then come to our energetics part. We already know we are producing 3 NADH, 1 FADH, and 1 GTP. Now, during the oxidation catalyzed by dehydrogenase enzymes of citric acid cycle, 3 molecules of NADH, 1 FADH2 are produced, and for each molecule of acetyl-CoA catabolized in 1 revolution of the cycle. So if you're talking of 1 molecule of glucose there, it already has produced 2 molecules of pyruvate. Eventually that converts into 2 acetyl-CoAs, which means 1 molecule of glucose would undergo the TCA cycle 2 times for complete breakdown. So in that sense, whatever we are calculating here, again needs to be multiplied by 2. These reducing equivalents are transferred to the respiratory chain in the inner mitochondrial membrane, which then undergoes that formation of oxidative phosphorylation. Now, during the process, we already know in oxidative phosphorylation, NADH generates 3 or a 2.5. I leave it at both again. I'll not conclude at one. Ideally, put that in your note. So, by esterification of ADP to ATP, and the process is oxidative phosphorylation. Now, again, the same problem would continue for FADH as well. All these years, our calculation was at 2 FADH produced at the end of oxidative phosphorylation. That no longer holds good. It's now believed that it is a little lesser than that, and that is 1.5. So again, our calculation there as well changes marginally. As per the old calculation, we always believed that one cycle of TCA would eventually give you about 12 ATPs, and that was the earlier calculation. Isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, one substrate level at succinate thiokinase or co-synthetase, whatever way we name it. Succinate dehydrogenase produced an FADH, which earlier was considered as 2. Malate dehydrogenase, the last but one step, would produce 3 ATPs, eventually giving you 12 ATPs in one cycle. But that we are now no longer holding good. So you can represent it diagrammatically. You can write the name and write the number of ATPs. It's up to you. How do you want to represent it? Diagrammatically also is good enough when you're writing a TCA cycle, because it will show you which step is producing the NADH or FADH. So even that is sufficient when you're writing energetics. Now this is from Vasudevan again, where it shows old and new calculation. A little complex in that sense if you want to write like this. So that's not required that you make such a big tabular column and write the energetics. Not at all required. You need to write one type of numbers, ideally or old or a new. Name the enzyme. Which step and all is not required, but just if you remember, whenever you're writing TCA cycle, even if they have not asked, TCA cycles energetics you can write, but just make a mention of a complete breakdown of a glucose molecule beginning with glycolysis. Though that was not part of the question, it's ideally understood and better if you have put that point additionally in your answer. Now, what does that translate to? Now, we began with those 2 steps in glycolysis. Hexokinase, phosphofructokinase. What was the calculation there? It was a -1. We are talking of 2 ATPs being utilized in the entire process. And hexokinase and phosphofructokinase, step 1 and step 3 of glycolysis, was the one that was a minus. So -1 in both those steps. Glyceraldehyde 3-phosphate dehydrogenase, all the dehydrogenase enzymes produce some NADH or FADH. Glyceraldehyde 3-phosphate dehydrogenase in glycolysis produce the first NADH, and that translated to 5 ATPs. Now all this we are multiplying into 2 from now onwards. So 1,3-bisphosphoglycerate kinase substrate level produced 1 ATP, which is multiplied into 2. Pyruvate kinase produced another ATP, which is multiplied into 2. Then don't forget this part of the calculation. This is the conversion of pyruvate to acetyl-CoA. Technically, it doesn't fall under glycolysis, neither does it fall under TCA cycle. Because when we are talking of TCA cycle, we don't have to write about this step, pyruvate to acetyl-CoA. When we are writing glycolysis, you don't have to write pyruvate to acetyl-CoA again, because you can stop your cycle at pyruvate. So therefore, this one step produces about 5 ATPs, but generally neither in the two pathways. So pyruvate dehydrogenase complex, again, it's not necessarily one enzyme, it's a combination of 2 or 3 reactions which is giving you an NADH which is multiplied by 2, and that adds up to another 5. Then in TCA cycle you had the 2 dehydrogenase, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase, both of them producing NADH multiplied into 2. Succinate thiokinase enzyme again produced a substrate level GTP. Succinate dehydrogenase and malate dehydrogenase produced one of them FADH and one of them NADH, which translates to 3 and 5. Under the old calculations, we had 7 and 8 for glycolysis, 8 as per old and 7 as per new. Pyruvate dehydrogenase would give you a 5 in case of the present calculation, 6 in case of old calculation. Generation in the TCA cycle per se, old I said it was 12, but now if you translate that, it is 10. So multiply by 2, there is a difference of 4 ATPs between old and new calculations. And net generation, as per the old calculation, it was 38. As per the new calculation, it is 32. So net number, you can write it at 32. Now, when the question is asked, it generally will not be a combination of the two. So therefore, if it is TCA cycle, just explain about these enzymes, name of those enzymes, how many ATPs it produced, how much a TCA cycle produced. You can write it as 10 multiplied by 2, 20 ATPs. And then for complete oxidation, you don't have to explain again with the name of the enzymes, but you write the total number of ATPs that are produced in old and new calculation. That is more than sufficient. A brief mention about that in the lower part would be sufficient for your answer. So energetics is an important part. Some give it almost 2.5 marks in a 10-mark question. So therefore, give some importance to it. Just don't write it in one word or one sentence. It is preferable that you really put some effort in explaining the energetics part, because eventually the significance of this entire breakdown from glucose until now was actually the ATPs that are produced. We are not bothered much about the products, intermediates formed, but the total ATPs. But one other important thing about TCA cycle, apart from the ATPs, are some of its intermediates. Now these intermediates, like we told, it's an amphibolic pathway, which meant it is both a catabolic and an anabolic. So we are also concerned about that anabolic process. We can't leave a TCA cycle just by saying it is just producing energy or ATPs. That's not the only reason a TCA cycle is happening. It's a step where from different metabolic pathways we get closer to the combined pathway, or maybe a central piece of that pathway, and we'll talk about that in amphibolic role. This you need not explain, but this question can be asked where the role of vitamins is seen in specifically B complex group of vitamins is seen in TCA cycle. Now this again will be explained on the chapter on vitamins, where B complex vitamins are taken and detailed. Each of them are dealt with, where you have riboflavin, niacin, thiamine, all of them as short notes. Each of these will be dealt with, where the functions of these vitamins will be dealt. So 4 of the B complex group of vitamins have precise roles in functioning of the TCA cycle, where in riboflavin, as FAD, alpha ketoglutarate dehydrogenase, and succinate dehydrogenase, both of these enzymes would have a role. Niacin as NAD, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, malate dehydrogenase, thiamine as TPP, where our decarboxylation reaction, alpha ketoglutarate dehydrogenase, had a role. TPP has a very significant role. Deficiency of thiamine can in fact even entirely stop the process of TCA cycle can cause multiple problems, whether in glycolysis or in TCA cycle, and can be a major hindrance. Pantothenic acid, again, as CoA, cofactor attached to acetyl-CoA and succinyl-CoA, each of these functions will be dealt with again in greater detail when we are talking of these B complex group of vitamins. So it's a combination question from vitamins and from TCA cycle. So I just put it here because a question like this can appear as a role of these vitamins in TCA cycle. Each of them are important. First and foremost, how is it regulated? The same as glycolysis. Need of ATP. That's the central piece. Do we need more ATPs or not? That will be the controlling factor of TCA cycle. Now, when the energy charge of the cell is low, as indicated by high levels of NAD or FAD, the cycle operates at a faster rate, so that it is converting to NADH and FADH, and that enters the oxidative phosphorylation and produces more of ATPs, and the cell gets enough energy. But that's not the only factor. There are certain other factors that are involved in maintaining a TCA cycle per se. The most likely sites for regulations are the non-equilibrium reactions, wherever there is a shift, and there are other factors that play a role in the entire regulation. There are occasions when these intermediates of TCA cycle are used up by some other pathways, either for the synthesis of, say, fatty acids, because acetyl-CoA is another very important link between fats and carbohydrates that can be diverted in some other pathways. We'll just see about that entire process called anaplerotic reactions. We'll just come to that, where we are talking about how these intermediates of TCA cycle are used up by some other pathways. In terms of the ATPs, which are the enzymes that are largely controlling it, pyruvate dehydrogenase, the initial conversion to acetyl-CoA, citrate synthase, NAD-linked isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase are the key equilibrium reactions which are maintaining the flow of TCA cycle either faster or to a slower rate. So the regulation part, we are concentrating on the central piece of formation of NADH and FADH depending on the need of energy by the cells, and that would be the regulation that we could be concentrating on. Pyruvate dehydrogenase, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. These are the key enzymes which are producing those ATPs, or rather the oxidative phosphorylation products. This is further explanation of it, where we are talking of which are the activators, which can be the inhibitors. When we're talking of inhibitors, we mean if there is enough and more production of so, we can inhibit the pathway. For example, if there is enough acetyl-CoA, more amount of acetyl-CoA, it in turn can inhibit the pathway. We have already produced enough acetyl-CoA, there is enough acetyl-CoA to enter the TCA cycle and produce those energies. If the numbers of acetyl-CoA, NADH or ATP is more, it can inhibit the pathway. If the ADP on the other hand is more and we need more of ATP, it can be an activator. So in the context of regulation, each of those pathways, each of those enzymes in the pathway can have an activator or an inhibitor. Largely, many of them will have inhibitors in the sense, because if that product is already there in the system slows down the pathway because the product is already there. We don't need more of energy there or ATP. Generally, in that sense, acetyl-CoA could be another inhibitor, because that's the one that is entering the TCA cycle and eventually producing those NADH and FADH. So these four, you can write it. It's up to you whether think there's another diagram. Yeah, this is the diagram. It's a little complex. If you want to, you can explain. We are just talking of what the inhibitors could be, what the activators could be. So each of those enzymes, I entirely leave it to you. The textbooks give you good explanation on each of these steps or each of these enzymes as to how they regulate pathways. If you are capable of dissecting it even further and understanding it, it's up to you how much you can explain for regulation. But regulation again will not carry more than a 2, 2.5 or a 3 marks at best. Very rarely 3 marks. 2, 2.5 is where it can be there. If you name the enzymes already, which are the enzymes and how it is regulated, it more or less carries more than 50% of the marks. Then in detail, if you want to explain about which could be the activator, which could be the inhibitor, it's entirely up to you. It is just further accelerating your or increasing the marks. So we told about that amphibolic role. Amphipathic role is largely to do with both catabolic and anabolic pathways and related to TCA cycle. Many times the question itself is framed when they want to ask you TCA cycle, they ask about this, explain about the amphibolic role of TCA cycle. So the question itself is framed in that manner. So you have to write about both the catabolic and the anabolic. It is about the TCA cycle. Nothing, no additional steps would be necessary, but we just need to understand which pathway, which products in this pathway are producing anabolic or required for anabolic synthesis of some other substances. So the main pathways include gluconeogenesis. Production of new glucose is possible from this intermediates, transamination, deamination, and fatty acid synthesis. So we directly can enter into the fatty acid synthesis mechanism, which will be dealt next in lipid metabolism, from where this pathway can be a starting product. Which are those intermediates that we are talking about? Oxaloacetate, the first step in this pathway, can start a gluconeogenesis, can be the substrate for gluconeogenesis. Formation of new glucose can happen from oxaloacetate. Fatty acid synthesis can happen from citrate, the first product that we had in this TCA cycle. Citrate can be the substrate for fatty acid synthesis. Now catabolic reactions, we have already seen that transamination and deamination, where alpha ketoglutarate and all those succinyl-CoAs were all involved. Deamination reactions as well, we saw that in the cycle itself. So both anabolic and a catabolic. We need to understand this a little further. Now this amphibolic role of TCA cycle or citric acid cycle, this pathway is utilized for both catabolic and anabolic to generate energy, as well as for anabolic reactions to generate metabolic intermediates for biosynthesis. So two important reasons why it's called an amphibolic. One, it produces energy. That was what we all saw it until now. The second one is it generates metabolic intermediates for biosynthesis. We saw fatty acid synthesis can take place from citrate. If the citric acid cycle intermediates are used for synthetic reactions, they are replenished by anaplerotic reactions in the cells. Now, if, for example, an intermediate, instead of getting into the TCA cycle, goes into the production of fatty acid synthesis, then what would happen to the TCA cycle per se? This would stop, or rather slow down. So we need an alternate mechanism in which, if it was used for anabolic process, these intermediates need to be filled up. And there are alternate steps for this. And this is the second part of the question in TCA cycle, where two words beginning with A, very often students get confused with the two words. One is amphibolic role of TCA cycle. The other is anaplerotic reactions of TCA cycle. Now, amphibolic and anaplerotic have a connection, but that's not the same. I hope you got the point. We are talking of amphibolic role. We used this as the explanation for it. It is a reaction that is producing energy. Also, it is that same TCA cycle produces metabolic intermediates for biosynthesis, and we need to give example for that, citrate or other fatty acid synthesis and others. But anaplerosis is another process where we are saying because of this amphibolic role, the intermediates of TCA cycle may be used up, eventually slowing down the TCA cycle per se. To overcome this problem, you have this anaplerotic reactions, and that is understood. Now this is a complex diagram to first explain, but just don't concentrate on other parts of it. Just take a look at these. Oxaloacetate, we said, can be used up for gluconeogenesis, but there are alternate steps which can fill up and produce oxaloacetate. You have to give at least 2 or 3 examples for anaplerotic reaction, and that is replenishing these intermediates with other pathways. Phosphoenol pyruvate, with the help of phosphoenol pyruvate carboxykinase, produces oxaloacetate. There is an phosphoenol pyruvate is a mechanism in which this carboxylase enzyme produces oxaloacetate, because this oxaloacetate was probably used for some other pathway. There are many other intermediates in the pathway, like citrate, for example, is used up for fatty acid synthesis and formation of steroids and other things. You have alpha ketoglutarate, which can be converted to glutamate, and it can enter into the formation of glutamine or certain amino acids, or even purines. You have succinyl-CoA, which combines with glycine to produce heme or porphyrins. Intermediate of TCA cycle, but enters into formation of heme. So there are a few of these important. Oxaloacetate has got the most important role here. It can enter maybe into glucose or even an aspartate. Pyrimidines can be formed from this. Phosphoenol pyruvate can go on to form serine, glycine, and a few other important amino acids. See the interrelationship of this one pathway in multiple different pathways of our body, from the formation of heme to fatty acids to steroids to purines to pyrimidines. You have a different mechanism in which these intermediates could have been used up there, for which we have a safety mechanism called the anaplerosis or anaplerotic reactions. So this citric acid cycle provides few precursors for certain biosynthetic reactions. You need to explain what is anaplerosis. Due to this contribution, there is a loss of certain intermediates of the cycle. To counterbalance this loss, we have what is called as the filling up reactions, which are called anaplerotic reactions or replenishing reactions. You don't need to give, there are many complex reactions that are involved, which are 2-step, 3-step processes, which eventually can produce either an oxaloacetate or a citrate, but we are not bothered much about it. We need to give 2 or 3 examples where this intermediates are formed from other replenishing or filling up reactions, because this is very often asked in the examination. Of this 6, at least 2 or 3 remember to write. One example you can give for oxaloacetate, or maybe you can give 2 examples for oxaloacetate, 1 for malate. This is replenishing. This is not part of the TCA cycle. We are only producing these products which are part of the intermediates of the TCA cycle. I hope this point is understood well. We were talking of these intermediates used up for some other biosynthesis. If it is entirely used up for biosynthesis, then what happens to this cycle? To make it viable for this cycle to continue, you have this replenishing or the anaplerotic reactions. If you know 2 or 3 examples from here, more than sufficient. Again, if you know with the enzyme, preferable, but even if you miss out, you should know that this intermediate can be formed from here. So pyruvate with the help of a ATP. Here we are not bothered much about energy consumption and other things, because we are talking of the intermediate to be replenished. So there could be loss of ATPs here as well, because the formation of oxaloacetate is using up an ATP. Pyruvate carboxylase is the enzyme. It produces oxaloacetate, but that's in a condition when this oxaloacetate was used up for other reactions. So it's a replenishing reaction. You can then phosphoenol pyruvate with the help of a GDP will produce oxaloacetate again. This is phosphoenol pyruvate carboxylase enzyme, which I gave you that time. There can be an alpha ketoglutarate or a succinyl-CoA. For example, in conditions where the succinyl-CoA is used up more for the formation of heme, you have a methyl malonyl-CoA. Methyl malonyl-CoA mutase enzyme will produce succinyl-CoA. Succinyl-CoA is also susceptible for being used up by for the formation of heme. Succinyl-CoA combines with glycine, an amino acid, to produce the first step in the formation of heme. So this is an intermediate of TCA cycle, succinyl-CoA. If used up in that, you need a replenishing mechanism, and that happens through a methyl malonyl-CoA. You can use 2 or 3 examples from here for giving anaplerotic reactions. Anaplerotic reactions has been asked 2 or 3 times before. It is either a part of the TCA cycle or can be asked separately with examples. So you can pick up, I'm not sure, this is probably from Harpub, where this diagram, you don't need to write such a complex diagram. You just need to understand that the intermediates can be used up, give examples for the intermediates being used up for biosynthesis, that biosynthesis being replaced by 2 or 3 reactions. Just further examples to this whole part itself. So this is what we have come to do in the last 3-4 hours, where we spoke about glucose breaking down in glycolysis, the intermediates of this were either used up in the formation of nucleotides. You could have a number of amino acids being formed, lipid mechanisms. You had a few important amino acids. The formation of TCA cycle, on the other hand, from heme to fatty acids to steroids, cholesterol, number of them are produced from the intermediates of TCA cycle, and therefore this entire pathway is more or less a backbone of many of the metabolic pathways that we will be coming across from now on. So there is an interrelationship that is built up here as well, and that will continue in the other pathways that we'll be taking from now on. I don't think I'll be able to complete this entire, but I'll just go through the significance. Now the problem with this pathway, HMP shunt pathway, I think every once in 2 or 3 examination, one question, you can 5-mark question that has appeared. I don't know how many times it has appeared now, so many times. Every 2 or 3 examinations, sometimes every examination, is significance of HMP shunt pathway. Till very recently, this was another pathway which I thought cannot be asked. Pathway per se, I'm saying. Significance, since a long, long time, it's always been asked. So one examiner probably thought I'll modify the question, and he gave the pathway itself, he or she. Now, I am of the opinion that this pathway per se is not very important. It is actually the significance that we want to know. The significance is important because of the 2 or 3 important products that are formed from this pathway. It is just a bifurcation of the glycolytic pathway, but what we need to know is the significance of this pathway. So I'll be dealing with the significance, the pathway. If possible, next week when there's some time, I'll try and finish it. Otherwise, you can read it on your own. There is a wonderful diagram which comes in Satyanarayan. I have that in my slides, where I think is a good enough, because here enzymes, formation of ATPs, NADH and all is not there. Enzymes are not very significant. There are 2 or 3 enzymes that regulate this pathway which could be important, but otherwise this pathway per se, I hope, is not repeated. But what has been repeated multiple times is the significance, and that you can expect in even the sessional exams. Most of the sessional exams will contain HMP path, significance of HMP shunt pathway. The other name, pentose phosphate pathway, is an alternate route of metabolism for glucose, where about 10% of the glucose molecules are metabolized. We had the glycolytic pathway reaching the TCA cycle, but this is just a shunt pathway. Why is this so important? The glucose 6-phosphate, a hexose monophosphate, instead of going through the glycolytic pathway, is shunted through this pathway, so it is known as the hexose monophosphate shunt pathway. Hexose monophosphate shunt pathway, HMP shunt pathway. This is because the glucose to the glucose 6-phosphate, instead of continuing in the glycolytic pathway, enters through the HMP shunt pathway. And what does its importance, what is its importance there? This glucose 6-phosphate has a capacity to produce very important intermediates.